Okay, can you see my screen? Neisseriae topic.
Yes.
Okay, good morning, BDSB fam, or rather good afternoon. Yeah, so we are going to have this discussion on Neisseriae. Sorry, I'll be brief. I have truncated it because of time. And I will send you a more comprehensive set of notes. Okay, but I want to point out what is important. And this is because afternoon, we don't want to rush you through the practicals. You have quite a bit. This afternoon, you should cover Staphylococci, Streptococci, and Neisseriae. So basically, you'll do practicals related to the cocci bacteria. So Neisseriae is a family that's made up of several genera. The most important one, the most important one, or rather our focus will be Neisseriae, something small on Branhamella, which has been reclassified. Some species have been reclassified into Moraxella. Okay. Okay, so this family, as you've seen, Neisseriae belongs to Neisseriae, and some important properties. One, morphologically, they are gram-negative oval cocci, and they occur in pairs. Okay, just like Streptococcus pneumoniae, which occurs in pairs, but these ones have a kidney bean shape. Strep pneumo have a lanceolate shape. And the two important pathogenic groups that we look at are the meningococcus and the gonococcus group. Okay, when you look at a clinical specimen, which you'd make from a gram stain from CSF specimen or from discharge in a case of gonorrhea patient, you'd notice that the bacterial pathogens are found inside the polymorphonuclear parcels. Okay. So this is what I'm talking about, the intracellular location of the bacteria within the polymorphonuclear parcels. Okay, from this, if you get a CSF specimen, a patient meningitis suspect, and you get such a picture, something like CSF is a sterile body tissue. So presence of this gram-negative diplococci is a preliminary diagnosis for meningococcal meningitis. Okay, so clinical specimen gram stain from clinical specimen is an important pointer, even as you wait for your culture results. Okay. Just a minute. All right. So still looking at some important properties. These are gram-negative bacteria. As you know, gram-negative bacteria have both outer and inner membranes. So Neisseriae have an endotoxin in their outer membrane that contributes to some of the pathogenesis that you see displayed in a patient with meningococcal meningitis or gonorrhea. Okay, some structural difference. For meningitidis species, it's LPS, while for gonorrhea species, it is LOS. Both organisms are inhibited by toxic metals and fatty acids on routine media. So the best media that you'd use for growing Neisseriae species would be chocolate, blood agar, and there are modifications of this based on the specimen. Like for high vaginal swab specimen, when you want to isolate Neisseriae gonorrhea, you use thiamatine media, which is basically chocolate agar potentiated with antimicrobial agents to suppress growth of normal flora that could have been also been collected when you are taking the swab from the patient. So these organisms, the clinically relevant species, produce this enzyme cytochrome C, which makes them oxidase positive. That's one characteristic for identification in the laboratory. For meningitidis species, it has a polysaccharide capsule that enhances its virulence because of its antiphagocitic nature. Both organisms may bear fimbriae. I'm saying may because we have certain virulent strains. Within the two species, there are certain strains that are virulent. Okay, those strains usually don't have fimbriae. And the gonococcal strains that can cause disseminated infections are found to bear numerous fimbriae that play important roles as far as attachment is concerned, gene transfer. There's the twitching motility type of movement that you see in those gonococcal strains, etc. But I want you to appreciate that the fimbriae play a very, very important role for virulence for both organisms. You can identify the species based on carbohydrate utilization, among other tests. Like Neisseriae meningitidis will ferment glucose and maltose, while Neisseria gonorrhoeae will ferment glucose only. So besides the two species that we'll focus on, the gonococcus and the meningococcus, just know that there are other members of that genus Neisseria that are commensals, common commensals of the upper respiratory tract, and some of them have been listed here. Now, Neisseria meningitidis will begin with this species. Human beings are the only natural hosts for meningococci, and people who are carriers of this bacteria have them in the upper respiratory tract. So the mode of transmission is by airborne droplets. Carriers, of course, are asymptomatic, and you tend to have a higher carrier rate in people who are in close quarters. For example, in prisons, in military training camps, in dormitories, hostels, etc. So because of the close proximity and the mode of transmission, you have a higher carrier rate in such populations. So from the nasopharynx, the organism can enter the bloodstream and spread to specific sites. More specifically, meninges. It can also move on to the joints and other organs when there's meningococcemia. Okay, so this picture is illustrating the meningitis belt in Africa that shows you some countries in Africa, and this belt also spreads to some countries in Asia where we have periodic outbreaks of meningococcal meningitis. Okay, although the incidence has come down because of rollout of several vaccination programs. Okay, so Neisseria meningitidis ranks second to strep pneumo as a cause of bacterial meningitis. Looking at the virulence factors for meningitidis, a very important one is the capsule, and this capsule enables the bacteria to resist phagocytosis by polymorphonuclear leukocytes. The capsule is the basis for classification of the serologic group. So we have serotypes given alphabetical letters under this species, A, B, C, D, E, all the way to Z. And those serogroups or serotypes are based on the capsular antigen. Okay, you can actually detect this capsular antigen in spinal fluid, and that is the basis for rapid laboratory diagnosis. And the vaccines that are available are also made using the capsular antigens. So that is the capsule. Other virulence factors include the endotoxin, which is responsible for these clinical symptoms that you see, fever, shock, disseminated intravascular coagulation, etc. We also have immunoglobulin A, IgA protease, which is an enzyme that cleaves secretory IgA. This is one of the first-line host defense mechanisms that you'd get along mucosal cells. So once this IgA is broken down, this enables the bacteria to attach to the membranes and invade. Okay, so it's one of the enzymes that are produced that facilitate invasion of the pathogen, okay, into the host. So what is the immune response? Resistance to disease correlates with presence of antibodies to the capsular polysaccharide. Any immunity that is mounted is serogroup specific. So protection against one group can still leave one susceptible to other groups, and people with complement deficiencies, any deficiency in their complement system, have an increased incidence of meningococcal bacteremia. Okay, so as I said, these serogroups are based on the lipopolysaccharide capsular antigen, and you can identify the serogroup you're dealing with using slide agglutination tests. So there are kits that are available. These kits have group-specific antisera. So you just need to do an antigen antibody agglutination test, which you can do on a glass slide, and where you have agglutination, you can identify the serogroup that has been isolated. Okay, and I told you we use alphabetical letters to designate the serogroup. Now, these serogroups are distributed differently. We have certain serogroups that are more prevalent in certain geographic regions. Some serogroups have been associated with people with certain underlying conditions like cancers or immunosuppressive conditions, etc. It is important always to know which serogroup is causing an outbreak. If there's an outbreak, you're a healthcare worker going there for an intervention, because any vaccine that you get will be serogroup specific. So identification of a serogroup is very, very important as far as initiating any intervention measures using vaccinations. So clinical manifestations, two important manifestations: meningitis, which can present as fever, headache, stiff neck, and the examination of CSF fluid will show that it's quite turbid because of the pus cells. For meningococcemia, which is dissemination once the bacteria get into blood, some of the clinical manifestations include fever, shock, DIC, thrombocytopenia, and adrenal insufficiency. Okay, so this is just a summary of the clinical manifestations. Some patients come down with a rash when they get meningococcemia, a rash that is described as a purpuric rash, the rash of meningococcemia. Okay, once you have the bacteria in the blood, the pathogen in the blood, you can have manifestations in other parts of the body or in different organs, as have been documented here in this table. So to make a lab diagnosis of meningococcal meningitis, this is the basic outline you'd use. And just to mention that this is an outline used for most laboratory diagnosis approach, where your goal is to isolate that pathogen and to identify the isolate from the clinical specimen. Okay, 98% of the time, this is the outline we use. I'm saying 98% because there are instances where we use other stains other than the gram stain. For example, processing sputum specimen for TB diagnosis, you'd use the ZN stain or any other stain. For Corynebacterium diphtheria, you'd use Albert stain. But for most, as you'll notice, most bacterial diseases, when you're looking at the clinical specimen, the gram stain is very, very important. So we can fill this in accordingly. The specimen that you'd pick would be blood in meningococcemia. You can take CSF specimen, which is the primary specimen for meningitis suspect, and any other specimen from those disseminated sites that I've talked about, but are not so rare. Okay, once you get your specimen, if it is CSF, you can centrifuge it, examine the deposit by staining it with gram stain and looking out for the gram-negative diplococci inside the polymorphonuclear pus cells. Okay, so that is what you'd see from the highly turbid specimen. Of course, part of the analysis, there's another arm that is carrying out biochemical analysis, clinical chemistry analysis of the CSF specimen, but our focus here is just on the microbiological lab processing. You can also culture the specimen after preparing your gram stain. You can culture for the blood in blood culture bottles, which you'll subculture to CBA. And for the CSF, you can culture it directly onto chocolate blood agar. How you obtain your specimen is very, very important. Remember, both blood and CSF specimen are sterile body tissue. So any isolate from such specimen must be followed to their logical conclusion. So you don't want to contaminate your specimen with normal flora from the skin because of poor disinfection techniques, okay, poor sterilization techniques. Then you give a report that is not accurate. Okay, so make sure you are observing aseptic techniques as you obtain this specimen. You culture them 35 to 37 hours, 18 to 24 hours in an increased CO2 atmosphere, like using a CO2 jar. You will be able to isolate your Neisseriae. Okay, so this is an example of a blood culture bottle. There are others that are available. This is just one of them, and this is the CO2 jar. After 24 hours, this is the culture plates that you'd get of Neisseria meningitidis on chocolate blood agar. Description of the colonies, small translucent gray colonies, and a gram stain of those colonies. You'll be able to see the gram-negative diplococci occurring in pairs. If you look at this keenly or closely, you'll see the pairs, okay? The pairs of this pathogen. So confirmatory biochemical tests, positive oxidase test, and fermentation of glucose and maltose. All right, just to distinguish between these two species so far, Neisseria gonorrhea, which we'll look at next, does not have a capsule, okay? And Neisseria gonorrhea ferments glucose only, unlike this species that ferments both glucose and maltose. Now, besides that long process test that would take 18 to 24 or 48 hours, other rapid tests that are available include countercurrent electrophoresis, latex agglutination, and PCR, depending on the equipment that is available. And of course, these are generally more costly, but they are better in terms of quick diagnosis, faster diagnosis. All right. For treatment and control, intravenous penicillin, cefotaxime, or ceftriaxone, it's good to use agents that readily cross the meninges when inflammation is present. Meningococci of group A and epidemic strains of group B are commonly resistant to sulfonamide, so these are no longer used. Chemoprophylaxis can be used to control outbreaks of the disease by eradicating the organism from carriers. And ciprofloxacin is one agent that has been used as a prophylactic agent where you give a single oral dose to adolescents and adults. This approach has been used in some countries where people are reporting to military training camps, recruits, or people getting into prison, etc. You're trying to eradicate the organism from carriers to reduce chances of getting an outbreak within that institution. Of course, vaccines are an important arm for prevention, and these vaccines contain the group-specific capsular polysaccharide. Please know that group protection is serogroup specific. There are certain serogroup capsular antigens that give a very, very good antibody response. Others, like serogroup B, require an adjuvant to elicit a good response. But your goal, of course, is to have the body produce antibodies to protect you, okay? The second species is Neisseriae gonorrhoeae, which is a bacterial pathogen responsible for causing an STI called gonorrhea. Okay, so gonorrhea or Neisseriae gonorrhoeae is exclusively a human pathogen. No reports of zoonotic infections, although chimpanzees have been infected artificially. A lot of studies are going on trying to come up with vaccines against this pathogen, Neisseriae gonorrhoeae, although those studies are still in the clinical trial phases. Where they're using animal models, they have used higher primates like chimpanzees, okay, to study the host's the host's antibody response using the vaccines. So the mode of transmission, this is an STI, so transmitted sexually and also during birth, as the child or the baby passes through the vaginal canal of a female who has active infection, presence of discharge, the baby can get infected when the mucous membrane from the eye gets into contact with the discharge from the mother. So manifestations of infection would be genital tract infections, anorectal infections for MSMs or people who engage in anal sex, or pharyngeal infections have also been reported for individuals who have oral sexual relations with somebody who has gonorrhea infection. So this is something that you'd not think about, at first instance, somebody who has throat infection, but these are things that have been documented and you need to look out for that. You can do a gram stain and you'll be able to see the gram-negative diplococci, okay, from the swab. So important virulence factors for Neisseriae gonorrhoeae include the pili, which mediates attachment to mucosal cells. There are some pili types that are also antiphagocytic. Okay, the lipo-oligosaccharide, which is an endotoxin, and outer membrane proteins. These outer membrane proteins play a very, very important role in immune evasion. They prevent establishment of an immunological memory. We have the IgA protease, which hydrolyzes secretory IgA, and porin A, which is a protein in the cell wall that inactivates the C3b component of the complement system. Okay, so this bacterial pathogen does not have any capsules. So gonococci primarily infect the mucosal surfaces in the genital tract, specifically the urethra and vagina, but you can have dissemination that would lead to manifestation in other parts of the body. And certain strains disseminate more than others due to their resistance to being killed by antibodies because of the kind of outer membrane proteins that they have, and also because of any form of deficiency in the complement arm of the immune system of that individual. Okay, so main host defenses against gonococci are IgA and IgG antibodies, the complement system, and neutrophils. Now, persons with a deficiency of the C6 to C9 components of the complement system are at risk for disseminating related gonorrheal infections. And repeated gonococcal infection is common primarily due to antigenic variation in the pili protein, okay? Antigenic changes in the pili and the outer membrane proteins. So antigenic variation, what is antigenic variation? It's variation in surface antigen composition as a mechanism of evasion of specific host immune responses. Okay, so Neisseriae does this by acids, by variants genes on off, and 3 horizontal exchange transformation of genes from other species. Okay, able to slide my it. Sorry, my internet went off. Can you see my screen?
Yes, we can see it now.
Okay. So I was just winding up on this antigenic variation that occurs in Neisseriae. And going on to the clinical manifestations, in women, asymptomatic carriage is common, and that is for a short time. Basically, what I'm saying there is that if you compare the male and female patient, because of urethritis in the male patient, an infected male individual would know quickly that there is an infection present, okay, compared to the female who may take a few more days before they detect that the infection is present. So for the female, infection is primarily in the endocervix, causing a purulent vaginal discharge. If it is not treated or completely resolved, gonococci ascend the fallopian tubes, leading to acute salpingitis, pelvic inflammatory disease with a high probability of sterility. Ectopic pregnancies can also occur because of formation of scar tissue on the fallopian tubes. In men, clinical manifestations include acute urethritis, dysuria, that's pain while passing urine. Purulent penile discharge is also common in the infected male. For the babies, a severe purulent eye discharge with periorbital edema occurs a few days after birth. This can result in blindness if it is not resolved. And you can use agents like 1% aqueous silver nitrate or topical erythromycin. Okay, just to point out, publications talk of 30 to 50% of infected females being co-infected with chlamydia trachomatis, which can also cause this condition called ophthalmia neonatorum in babies. So it's good to pick agents to when this baby is born and you notice the female has active discharge, probably Neisseriae gonorrhea related or chlamydia trachomatis. For the baby to take care of this condition or to prevent it, pick an agent that can take care of both chlamydia and Neisseriae. Okay. So that is a periorbital edema and the discharge from the baby's eyes. So dissemination, which I mentioned, can occur with certain strains or with people with deficiency in the complement system. Peritoneal spread occasionally occurs, and this may lead to perihepatic inflammation. Disseminated infection can also present in the joints. So there is gonorrhea, arthritis, fever, septic lesions on the skin, and rarely endocarditis. Okay, a patient who had several such lesions on the skin, and it turned out to be a case of disseminated gonorrhea. So for lab diagnosis, this is the outline that you'd use. You saw this in the previous slide. If we can fill that in, the specimen to pick for diagnosis of gonorrhea. For the male and female adults, okay, female patients, you can take urethral specimen from the male, high vaginal swab, or cervical exudates from the female. If you're not able to get a urethral swab from the male, you can ask for a fast voided urine specimen, which you'd need to centrifuge. Anorectal infections, a rectal swab, and eye swab from the babies with ophthalmia neonatora. A transport media to maintain viability of this specimen is Stuart transport media. Okay, so you need to use a urethral swab specimen that is specific for getting that specimen. It's not any other swab, okay? Because the urethral swab specimen, the urethral swab is small and appropriate for picking a urethral specimen. Other swabs may be a bit painful or uncomfortable. Okay, so that's the Stuart's transport media where you'd introduce your specimen to transport to the laboratory. So gram stain of the specimen in men, the finding of gram-negative diplococci within polymorphonuclear cells in the urethral discharge is sufficient for diagnosis, preliminary diagnosis, okay? But of course, absolute gold standard would be to isolate the pathogen from the specimen through culture. So in women, there's a higher risk, there's some risk of getting false positive and false negative reports from the gram stain alone. Okay, because when you take a swab from the female, it is likelihood of harvesting normal flora, okay? And so you may get that false negative due to inability to see the small numbers of cocci amongst the lactobacilli and other bacteria that are normal flora, okay? So for the urethral, it's easier to make a conclusive report from the urethral swab of the male compared to this swab from the female. However, both of them are important. Just the take-home message here is that you need to be very, very keen when you're looking at the swab from the female patient to make sure you don't lose, give a false negative because of the inability to see the cocci amongst the other bacteria. Okay, so you can see such a picture. Okay, swab, gram stain from a cervical swab. These are commensals like lactobacilli, and this here is a polymorphonuclear cell with the gram-negative diplococci. So if you're rushing, you may easily miss this and give a false negative report. So culture on CBA or on thiamatine media, thiamatine specifically useful for high vaginal swabs. This is a chocolate blood agar that has these antibiotics, which suppress these other normal flora that could possibly have been obtained while getting the specimen. These are the conditions for culture, significant that it grows well in increased CO2 atmosphere. So you put it in a CO2 jar. Okay, and to confirm identity, again, you can carry out oxidase test and glucose fermentation test. We also have rapid tests for gonorrheal diagnosis, ELISA kits that can detect gonococcal antigens, and DNA probe assays to detect gonococcal ribosomal genes. Okay, so for treatment and control, penicillin is no longer used. I'm just putting it here to point out that resistant strains have been isolated. They possess the gene coding for beta-lactamase, and this is really, really common. Okay, so fluoroquinolones, tetracycline, expanded spectrum cephalosporins, amoxicillin, clavulanate would be preferred. However, from the notes that I'll share with you that are elaborate, I will share with you the current single and dual therapy recommendation from WHO as treatment guidelines for Neisseriae gonorrhea because of a lot of resistance that has been reported. Okay, WHO has classified this organism as a priority 2 organism because of resistance. CDC classified it in 2012 as a superbug. So we have incidences or reports of gonorrhea that cannot be treated by any non-antimicrobial agent. Okay, that can be another epidemic or pandemic. Okay, so the notes that I'll share with you are elaborate as far as what is recommended for treatment of pharyngeal infections, for anorectal infections, etc., for prevention of ophthalmia neonatora in babies. A recent recommendation that was last year, 2020, by CDC, requested or recommended that people should revert to single therapy rather than using dual therapy. Dual therapy recommendation involves a combination of agents like ceftriaxone, and azithromycin. Okay, but we have recommendations of using single therapy to reduce chances of resistance to the two agents. Okay, so you will read about them, but the take-home message which I want to point out as far as resistance is concerned, that any isolate from a patient, okay, should be followed up by antimicrobial susceptibility tests, so that you're treating that particular patient with drugs that that pathogen is susceptible to. It's also good to be guided, because we don't have microbiology labs everywhere. So it's good to be guided by the local empirical guidelines using data that has been generated by laboratories that have looked at the AST profile of the pathogens that have been isolated locally, because naturally you'd expect what is circulating locally to guide you as far as treatment is concerned. But when you have a patient and you have this access to microbiology lab facility, please get that AST profile report for that particular pathogen. So prevention of gonorrhea using barrier methods like condom, of course, rapid diagnosis to treat the patient as well as their contacts, use of effective antibiotics based on the AST profile from the lab, okay, prompt tracing and treatment of symptomatic patients and their contacts. Some aspects of public health education would be involved here. In some jurisdictions, gonorrhea, being an STI, is a notifiable disease, and you're required legally to inform your sexual contacts so that they can get treatment. Otherwise, you'd be liable for some legal action, okay? We don't have vaccines that are available. Yet it's still elusive, but active research is going on. In the notes that I will share, I'm not going into that now because of time, but in the notes that I will share, I'm going to include some write-up on what is going on in terms of trying to come up with vaccines against gonorrhea, okay? But it's still under research. Okay, so this was a supplementary genus, just to point out what is important here. One is that classification has been done and reclassification. Some species that were classified as Branhamella were reclassified into Moraxella. Okay, they belong to this family Neisseriae. The two important pathogens here would be Moraxella catalis and Moraxella non-liquefaciens. Okay, so this first one is the major pathogen in this genus. It's only found in humans and transmitted by respiratory aerosols. So it is an upper respiratory tract commensal, but once it gains access to the lower respiratory tract, it can be pathogenic, and this can happen in patients who have chronic obstructive pulmonary disease or their ciliary motion is impaired. Okay, it has also been implicated in otitis media and sinusitis, primarily in children. So lab diagnosis, you can take a bronchoalveolar lavage specimen or a sputum specimen. On gram staining, you're going to see the large numbers of gram-negative diplococci dispersed between cells. Okay, you can culture it on blood agar or CBA, and it's positive for these two tests, butyrate esterase and deoxyribonuclease tests. For treatment, you can use trimethoprim, sulfamethoxazole, or amoxicillin clavulanate antimicrobial agents. The other species important in this genus is Moraxella non-liquefaciens, which commonly causes eyelid infections or blepharitis. The other common pathogen that causes eyelid infections or styes, commonly called styes, Staphylococcus aureus. Okay, so the treatment is local application of erythromycin antibiotic. Okay, so the rest are supplementary. You will read more about them: Acinetobacter and Veillonella. Okay, I have rushed through because some of you need to move to, maybe you're using Wi-Fi somewhere and you need to get to Kenyatta for your practicals. And I want to do a recap for this when we meet, okay? Because we are trying to reorganize your lectures and practical sessions. We have got feedback on challenges of having lectures practicals from 3:30 to 5. So we are trying to see what would work best for you, either having practicals 2 to 5 or alternating lectures, practicals, etc. So I didn't want to miss this session. The cultures had also been prepared for Neisseriae, so we didn't want that to go to waste. So I hope you have an idea, much as I have. We have gone through this quickly so that you're not lost during your practicals. This is an important bacterial pathogen, genus Neisseriae, because of those two conditions: meningitis caused by meningococcal meningitis, or Neisseria meningitidis bacteria, and gonorrhea, which is a sexually transmitted infection caused by Neisseria gonorrhea. Of major concern right now is resistance of gonorrhea to several antimicrobial agents. Okay, these are gram-negative diplococci, and you can identify them. You can grow them easily in the lab on chocolate blood agar. You can visualize them from clinical specimen, and you can carry out biochemical tests to identify them. Okay, when we'll meet next for our next session, I will have a recap on vaccines, the vaccine research going on Neisseria gonorrhea, okay, and other important aspects of Neisseria meningitidis. But I'm going to include all that recap material in the notes that I will share with you. Okay. So I'm going to release you for the afternoon so that you make your way to Kenyatta for those who are not there. I'll share the notes through your class rep, the more elaborate notes, and then we can have a recap. Please take care as you have your practicals, observe the social distancing and all recommendations that you're given. Thank you. Have a good afternoon.